Hi there, this is Dr. John Whitcomb talking about Mitochondria Primer Part 3, how it goes terribly wrong. And let me give you an example. We learned last week that mitochondria are the critical components that extract energy from carbohydrates by transporting protons and electrons across a membrane, like pumping water up a dam and then letting the protons come back across that membrane and extracting energy. But mitochondria, there's 200 or 300 of them in your muscle cells, but 5,000 in your heart cells and in your brain cells, and your other organs have various numbers of them. So you have a lot of them in each cell. Mitochondria have to have copies of their own DNA so that they can rapidly respond to rising and falling energy needs. It wouldn't do to be able to have a need for more proteins to help make more electron transport chains if you had to call all the way to the cell nucleus. That would take too long and be too indirect. So mitochondria have 10 or so copies of the most important DNA that they need, that they copy themselves. And that DNA, of a necessity, has to be close to the electron transport chains where this energy production is going on. Well, you could think of the electron transport chain as a roaring furnace, or you could also think of it as a zoo with some slightly loose cages. But we do know that some electrons escape the electron transport chain because electrons, the Heisenberg uncertainty principle says that electrons aren't always particles and they aren't always waves, and the poor proteins have to sort that out. So about 2% of electrons escape and get away. And when they do, they attach themselves to oxygen and they make reactive oxygen species, which are very damaging. If it damages DNA, you're going to get injured mitochondria. So how does it go wrong? Well, Thomas Seyfried, a PhD biologist at Yale, has made a CT scan of an electron microscope and has essentially shown that you can look inside the mitochondria of a cell, and instead of seeing nicely ordered membranes, it looks like curdled milk. Now, the ordered membranes have all the proteins of the electron transport chain in a good sequence, but the curdled milk, they're just all hodgepodge, and you have a dramatic reduction in the energy production ability of that cell. Now, what that means is that if you actually take mitochondria from a cancer cell, that's what passes on the cancer ability. You can take a normal cell and put the nucleus of a cancer cell in it, and it'll still be a normal cell. But if you take the mitochondria of a cancer cell and put them into a normal cell, it'll turn into a cancer cell. So what's passing on the quality of a cancer is the damaged mitochondria, the damaged DNA from the local damage happening from reactive oxygen species. So cancer isn't 500 different diseases. It's perhaps one disease, damaged mitochondria, and in 500 different tissues. Now, the level to which the mitochondria is damaged is the level to which the cancer cell can't burn fat. Well, isn't that interesting? If a cancer cell can't burn fat, but your regular cells can, what happens if you eat a lot of fat? You're leaving the cancer out to dry. And if we now knew that green vegetables actually get turned into short-chain fatty acids in your gut, then a diet for cancer would be a high vegetable, green vegetable, not roots, green vegetable diet. Because the only thing a cancer cell can live on is sugar. Lots of sugar. So we want you to avoid sugar, we want you to avoid carbs, we want you to avoid grains when you have cancer. That's how mitochondria can go terribly wrong. If we're going to cure cancer, we've got to delve deep into the defect of cancer, and Thomas Seyfried is the first to lead the way. Look up some of his YouTube videos. You'll be fascinated. He's on to something big. This is Dr. John Whitcomb ending Mitochondria Primer on how it goes terribly wrong.